Hi, my name is Joana Cabral, and I'm going to talk about resting state activity from the perspective of Dynamical Systems Theory. So my talk will, in the beginning, I will start by introducing a bit the concept of Dynamical Systems Theory and how we can look at the brain from the perspective of a dynamical system. And then I will introduce the concept of synchrony as opposed to phase locking and how this can be two different features to characterize stability in dynamical systems. Then, focusing in resting state brain dynamics, I will explore the concept of these resting state networks being ghost attractors, how they are expressed as transiently phase-locked patterns that emerge in brain activity that are recurrent, how we can characterize the dynamics in state space, and how this relates to psychopathology, how it can be important for psychiatry, for psychology, so understand psychophysiology in general. Then I will introduce the new recently released LIDA toolbox. I will show a bit how to open and run the codes and how which results can be obtained from it. Okay, so starting from dynamical systems, basically any system in nature that is not static but evolves over time can be approached as a dynamical system, as the name states. So these are systems of interacting units, can be atoms, molecules, cells, life forms, that can exhibit collective behavior that cannot be understood by the single unit alone. So there is a behavior that emerges, so the word emergence is very common in collective phenomena, at the macroscopic scale. So the theory of dynamical systems serves to search for the fundamental rules governing the collective dynamics emerging at the system level. Now, why does this happen? This emergent collective behavior emerges in systems where these patterns have a function. So these systems are termed complex adaptive systems because the collective has a shape, and this shape has a function that increases the chances of survival or of auto-sustainability, so of lasting longer. And this is observed across a wide range of scales and forms, so it's a universal concept, right? And there are tools from statistical physics that have been developed to characterize the general properties of dynamical systems to help decipher their governing rules. Okay, so for example, we can talk about stability analysis. So you can have just a dynamical system that is constantly in the same behavior. So it's just going up and down, up and down. So when I say oscillatory, it's like a pendulum or the orbit of a planet or something that constantly is mostly in that state. So we can say it has one stable state. It's not just a static point, it's not a fixed point attractor, it's an oscillatory attractor, so it's dynamic. It returns to it usually upon perturbation, so if we perturb a bit the system, it will return to this state. So we tell it has one basin of attraction. It is like a well of attraction, and it's called also an attractor. Now, if the same system, if you change a little bit external parameters or you perturb it in some way, it can switch to a different dynamical behavior, so a different regime, a different state of activity, let's say. And in this case, we say that the system has multi-stability. So here it's the same membrane, just as a general illustration. And when you perturb with different frequencies, it will respond with different patterns. So in this case, it has 3 attractors, but it can have more. And the thing is that the system switches between these attractors by external perturbations. Usually, even noise may drive transitions between these states. So then we can talk about trajectories. So imagine this membrane, it has this repertoire of attractor states that exist, they are possible, and imagine that time is represented by this green ball. So over time, the system may switch between these different attractors that exist, or these different states, substates that exist in the repertoire. So this is a way to characterize these trajectories of the system. There are tools to characterize, but one of the peculiarities of these systems with multi-stability is that they are sensitive to the minimal change in one of the units in the system can induce a transition to another state, right? And so another stable state, and so it can drastically change the behavior of the whole system. This is called the butterfly effect, in the sense that the flapping of a butterfly in the Amazon can change the weather in Asia or in other parts of the world. Okay, so this is an important concept. And then there are tools, metrics, let's say, to characterize the behavior of a system, of a dynamical system, from empirical observations. So we need to define what we consider to be a state, right? And then once we divide let's say we analyze the dynamics of a system, and then we can cluster it, the behavior, into different patterns of behavior. And then we can calculate what is the probability of staying in a given state. This kind of infers the stability of this state, how long you remain in this state as well. This is the dwell time. So once you enter in this state, how long do you remain there? Okay, it is related to the fractional occupancy, but doesn't capture, each of these metrics captures slightly different things. And then there's also the transition probability. It's also called the Markov chain of a system, which in a way is the probability of switching from one state to another, given the probability of being in a given state. Okay, so these transitions, it's assumed that it's probabilistic. And then if we find that some transitions are not possible, or are more possible in some conditions or in other observations, so then each experiment, so each scan, for example, of brain activity, is a different observation of the system. Of course, we cannot obtain a full characterization of all the possibilities, but during each session, we can compute these statistics. So now, why can we look at the brain as a dynamical system? Well, because we all agree that there's a structure, okay, that's composed by its units, the neurons, but also you can argue there are other elements, the glia, astrocytes, ions, fluids. But overall, this structure has activity patterns, and this nearly infinity of activity patterns that our brain can exhibit, that seems to be coordinating behavior. So indeed, over the last decades, there have been consistent reports of functionally relevant activity patterns that are detected with fMRI, with functional magnetic resonance imaging. So there are patterns of activity that appear during rest, but they are also constantly recruited during specific tasks, so they have a functional relevance, and that appear recurrently, transiently, consistently across scans, and we still don't have an intuition where they come from. What could they be? Expressions of attractor points in the brain? So from the perspective of statistical physics, these intrinsic connectivity networks, or resting state networks that have been reported in the literature, can be hypothesized to represent a repertoire of attractors. right? But which features do they exhibit? How far can we go into the characterization of these patterns? So here I'd like to start and introduce and make a small parenthesis to interpret the temporal signature of the activation of these patterns. So when we talk about activation, is this co-activation, which means like different areas that simultaneously increase their signal at the same time, but for a brief period of time without any specific periodicity? In this case, we just apply a threshold and can detect co-activation patterns. Another way to analyze and to detect these components is to look at the correlation of the signals. And the correlation implies that you need an interval of time. It can be the whole recording session or just a window. And we see if across this interval of time, how correlated the signals are over time. So going either up or down. So it doesn't matter just that they simultaneously pass a threshold. What matters is that they are going together. Now, if there is a periodicity in the signal, in the temporal signature, so if there is like a specific timing, we can talk about oscillations. And if the signal is indeed oscillatory, then the term to characterize when you have two signals that share the same oscillatory signature, we can say that they are synchronized. And they are synchronized in phase. If the two signals are exactly identical, so they overlap, so you can find that they go up and down at the same time, or they can be phase locked in the sense that it's the same oscillation, but it is shifted in phase. Okay, so these concepts of synchrony and phase locking have the implicit assumption that there is an oscillation driving this signal. Okay, and when we talk about oscillations, it's very interesting because there are many systems in nature exhibit this oscillatory behavior, and oscillatory modes do not necessarily need to be the whole system to be synchronized in phase. You can indeed observe phase locking, which means that some units go up while the others go down. So in some modes, these are called the antinodes, for example, of a standing wave pattern or a stationary wave pattern. And in one mode, like two voxels can be in antiphase, then when you switch to a different mode, as I illustrate here, the same voxels may be in phase. Okay, so there's no exclusivity that some voxels only belong to one node or one pattern. So the same nodes may be antinodes of different patterns. And this is the repertoire. So now this builds like an hypothesis of, are these intrinsic connectivity networks that we are detecting in the resting state the expression of phase locking modes in brain activity? And to analyze this, we need to analyze the phase of the signals that we are detecting. And assuming that there is an underlying oscillatory nature in the fMRI signal, which is typically detected in this range below 0.1 hertz, but we are finding consistently there are new reports showing that this extends probably up to slightly higher frequencies, up to 0.2 or 0.3 hertz. So we can take the signal in that range of frequencies, compute the Hilbert transform, which kind of creates an imaginary component in the signal from which we can then extract the phase and the amplitude of the signal. And the idea here in this analysis is, so we discard the amplitude, we don't care about going above a given threshold. What matters here is the phase of the signal. Okay, so we can have at each instant of time, we can see the phase of the signals distributed across the whole brain. So this is a picture, like a snapshot from one instant of time of the phases. And what matters is not the specific phase at which they are, but the phase relationships. So as you see here, as I surround here with the green circles, all these units, all these brain areas seem to be aligned in phase. So they are shared, they are represented with the same color. And if we want to capture just a phase relationship, we need to do the difference in phase, okay? And we do the cosine of the phase difference. So when the difference in phase is 0, the cosine of the phase difference is 1. Okay? And if they are in antiphase, which means when one goes up and the other goes down, okay, then the cosine of the phase difference is -1. So this is a measure to capture these phase relationships between units at each instant of time. And then the leading eigenvector of the phase coherence matrix is a vector that captures the dominant pattern of phase relationships. So it's very interesting because we can summarize this matrix of phase relationships by one vector. Actually, it's a rank 2 matrix, so it can be represented by the first 2 eigenvectors, because it's only there are only these two properties. But if we consider just a leading eigenvector, I will show that we get very relevant results. So this analysis is termed leading eigenvector dynamics analysis, and it was introduced in 2017. And the thing is that, as you see on the right side, this is the elements in the eigenvector. So each element represents a brane area. And the sign of the elements in the eigenvector tells us which is the main direction of the orientation of the phases, right? So the ones in blue are oriented in one direction with respect to the eigenvector, and the units that are represented in red means that they are projecting in the opposite direction with respect to the main orientation of the phases. And so it seems that there are some areas that are shifting in phase from the rest of the brain, as I illustrate here in red and blue. So now if we look at the phase coherence matrix at each instant of time and we characterize how it is evolving, we see that there are constantly different patterns that we can observe, right? And on the right side, what I'm doing is I'm plotting links between the units or the brain areas whose phase is shifting from the main orientation. And this may appear very random, but if we look closely, we find that there are some patterns that recurrently appear, that sometimes relate to the default mode network, sometimes more to the frontoparietal system. So there is some structure in this activity that is interesting to characterize. We apply some analysis. So the idea was to take, okay, at each instant of time, we have these eigenvectors. We save them all at each instant of time. So in a way, we transform the fMRI signals that we detect, we compute the signal phase, and then the leading eigenvector. So we have still n times time. It's the same size as the original parcellated signal. And now let's consider that each dot, so each eigenvector, here we obtain all the eigenvectors from 100 scans from the Human Connectome project. So for each volume, we compute the eigenvector. And then each one here, we compute the covariance matrix, we take the first 3 principal components, and we represent a cloud of observations in this reduced 3-dimensional space, where each dot represents one eigenvector, which is one pattern of phase relationships in the brain. And then we are going to cluster this, because we want to see if there are some recurrent patterns of activity that relate to the resting state networks, right? So we can partition this cloud into a growing number of K clusters, let's say. So here is the example for K equal 5. So if we tell the K-means algorithm to break this cloud into 5 clusters, it will run several iterations. Okay, we wait until it converges, but it will divide the system into 5 states. And we can do this for different Ks, right? So we typically do, for example, between 2 and 20 states, because we don't know exactly how many resting state networks exist in the brain. And here what I did is to overlap the centroids that are obtained. So these red regions, the areas in red, so the smallest community of units that is shifted in phase, with networks from the literature. Here I took the networks from Yeo et al. 2011, the 7 reference networks, or intrinsic connectivity networks that were defined for 1,000 subjects. And I use this as a reference to see if the patterns that we obtained by clustering the leading eigenvectors overlap with this, right? So if we just cut into two, if we divide into two, we find that one mode is the global mode, so everything is line in phase. There's no particular subsystem that is detaching. So it's represented by the transparent brane, and it's the most probable, the one that occurs with more probability. And then the second one here overlaps with the default mode network. As we see, as we increase the number of states, like up to k equals 6, the second most probable state is usually the default mode network. Then it becomes the third, the fourth, because then we start detecting other subsystems that are synchronized. But here I will show the results for k equal 5. So with k equal 5, we obtain the global, the default mode network, one that is yellow overlaps with the frontoparietal, the other with the visual, and then with the ventral attention somatomotor. Okay, and we can compute the silhouette value here is to find exactly what is the best number of states that represents our data. But this also has some peculiarities, and sometimes the best number of states is not necessarily the one that is more sensitive to the specific study in question. What I mean is, in some psychiatric disorders, for example, we may need a finer grained parcellation into, for example, 20 states to identify exactly which is the pattern that is going wrong in this disorder. Okay? So by looking here at the 5 states that we obtained, I represent with arrows how they project into the leading eigenvector, just to illustrate what this means. You see, it has this feature of the standing wave patterns of stationary waves, which means when the signal in some areas goes up, it goes down in others. And this is what is characterizing these patterns of activity. And we see here how they overlap with the different resting state networks from Yale, where the 5th one overlaps with both the somatomotor and the ventral attention. And then we calculate things like the fractional occupancy, which, as I said before, is the probability of a given pattern to occur over time, the dwell time, how many consecutive time points are assigned to a given state. And when we compare this, we found that there is reliability. So the same 100 subjects were scanned twice. And here what we are showing in for example, the fractional occupancy, on the y-axis it is the first session, and on the x-axis is the second session. And you see, for example, that the probability to be in the first state, the global mode, these probabilities are close to the line, to the diagonal. So this means that there is some reliability. The people who in the first scan spend more time in the global mode, in the second scan, the second day, they will also spend more time in the global mode. Also, what we find is that the other networks, and here the probabilities of the global mode have a lot of variability. In some people, they last like 70%, in others 20% of the time. Now, the probability of the other networks, you see, they're always less than 50% of the time. The DMN is the one that really reaches higher values, but we see, for example, the frontoparietal or the visual appear only like 20% of the time, so 0.2. consecutively across 100 scans, actually 200 scans, right? So they always occur briefly and they last shortly, as we show here with the mean dwell time. Okay, so they last around 2-3 seconds, which is the TR of the resolution that we have. So in a way, we don't have enough sufficient temporal resolution to characterize the temporal signature of these states. But one thing we can do is to characterize their transition probability, to see what are the trajectories in state space, right? And what we found is that there's consistently, so in the probability matrix of transition, you see that there's consistently like the transitions to transit back to state 1, from state 1, state 2, state 3, state 4, is always higher, is lighter blue, okay? So you always have like 20% probability to return to state 1. So the main attractor is this global mode of activity. Let's call it like a fundamental mode or a baseline mode or something in this perspective. And then from time to time, with like 8% probability, it will switch to the DMN. It has 50% probability to remain there in the next time point, and then it decays back to the fundamental mode. So that's why these other states that relate to functional networks can be approached to as ghost attractors, in the sense that you switch there, but they are not very, very stable. You approximate them, but constantly the system decays back to the fundamental mode. Okay, and this has relevance for psychiatry, because we are finding on different applications that these trajectories are changed in diseases, and also the occupancy. For example, in schizophrenia, there's highly significant reduced ability to remain in the fundamental mode, okay, in this global baseline state where everything is synchronized together, and there are significantly more excursions, so more activations or more switches to the visual network, to the somatomotor network, and to a limbic network. So this kind of reveals insights of that these psychiatric disorders may be related to dysfunctional trajectories in this state space. Okay? Because they are very specific. It's not just random, it's not just noise, it's specific to some specific networks and may relate. And I've applied this already. We found alterations in people in remission from major depressive disorder. There are also significant changes in people with obsessive compulsive disorder, so they spend significantly less time in a network that synchronizes the orbitofrontal cortex with the basal ganglia. Also, we found with trait self-reflectiveness that it relates with the probability of being in the DMN. Psychoactive drugs, for example, psilocybin was found to significantly suppress the probability of being in the frontoparietal system, which is the executive control network. Also, deep brain stimulation activates the orbitofrontal system, as well as music or watching smiling babies. So that there are different insights that I've been having by applying this tool, this leading eigenvector dynamics analysis. I've already applied to over 30 data sets from collaborations from all over the world, and also from open source data sets. And so far what I find is that these states of phase locking are very consistent. They are transient, they are short-lived, they are recurrent, they are meaningful, and they are reliable. Okay? And importantly, the dynamics or these trajectories in state space and the stability of these networks is altered in psychiatric conditions. It can be modulated by psychoactive compounds. Also stimulation, both electromagnetic stimulation but also visual or auditory stimulation can change the occupancy of these states. And they are also modulated by tasks and by preclinical features, in the sense when you're sad or when you're thinking more about yourself, or cognitive performance as well. So, but despite this evidence, the biophysical mechanisms underpinning brain functional networks remain unclear. And I think it's very important to gain further insights into the origin of these functional networks. And now let me briefly introduce to the first version of the LIDA package release, because so far it was always like sending emails with scripts and things. So now we put together a zip file, let's say, with the scripts running both in MATLAB and Python. So in MATLAB it was in collaboration with Miguel Farinha, and in Python with Álvaro de la Iglesia, who organized this. Then we also prepared tutorials to show how to run the scripts and adapt to your data set. And the figures are generated automatically, they are saved in a folder, and it's been optimized to compare fMRI scans across n conditions. So if you want to compare 3 conditions, 2 conditions, 4 conditions, but it then can be adapted to different paradigms or tasks, let's say. The full details and information are at the website psychomark.com. Okay, so overall what you need, you just download the zip file, you unzip the folder, you fill in the details of your data set, like the TR, the folder where the data is, the tags, the name tags that were given for the different conditions, and then the directory where you're saving, and then you press run. Okay, and if everything goes well, it will explore for a range of K. Okay, the networks that are obtained. You should obtain like this pyramid of centroids to see that they overlap with what has been reported in the literature. It compares also the probability across conditions and provides like an analysis, a statistical analysis correcting for multiple comparisons. Okay, we apply Bonferroni correction, and so far, like If it passes the Bonferroni correction, I trust the results and they've been quite meaningful. If it doesn't, I recommend to analyze again or to do other things, but this doesn't do magic. Okay, but usually we find that there are significant results here. What we are comparing is between a condition with DMT, is a psychoactive compound, compared to placebo, before and after. So when we compare before and after placebo, there are no differences. So here we see there are no asterisks below the blue line in the p-values. When we compare any condition like before DMT or before placebo with the DMT condition, there are highly significant changes. And then we can choose one K. For example, in the codes, you choose K equal 5, because it shows some results interesting, and it generates the plots for these 5 networks that were identified. Here, for example, we detect the global, then the second is the DMN, then the frontoparietal, then the somatomotor, and the visual. Okay, you recognize this a bit because it also was the same networks that came out in the Human Connectome project. Sometimes they are ordered differently. Because of the probabilities can change a little bit, but spatially there is a very strong overlap. And here we show, for example, that the probability, so it generates automatically also the plots for the statistics, so the global state increases during DMT and the frontoparietal network decreases. And these results are obtained in less than an hour, and they were fully aligned with the results that we published in 2019 with psilocybin, showing exactly the same effect, so a suppression of the executive control system. So, and there is some relationship because they both affect the serotonergic system. And then you can choose one specific centroid and analyze it in detail. You can sort, you can obtain the list of brain areas involved, you can look at the box plots for probabilities, dwell times, and represent into different formats. And so thank you. I hope you give it a try and go to the PsychoMark website, but also please don't hesitate to contact me. I will try as much as I can to give some support if you find errors. This is still ongoing process, of course. Special acknowledgements to Miguel and Alvaro who prepared the script, and also Jacob who contributed a lot to all the scripts and the images, Josefina who prepared the tutorials, Pedro Morgado, Gustavo and Morten for fully supporting this research. And also I invite you to come to my posters that I will be presenting here at OHBM. It's slightly unrelated subjects. I'm operating on different fronts, but I would be very happy to discuss any ideas that you find relevant, and thank you for your attention.